Bruce, it was a very interesting talk you gave this morning. What can you tell me about it?
Well, I talked about what are the prospects for now and the future in forensic genetics. As you know, forensic genetics helps us identify individuals, and that may help solve cases, can exonerate the people who've been wrongly associated with cases, the innocent, may help determine cause of manner of death. All these sort of different things that can help us assist in law enforcement to help solve crimes.
Yes, and you were talking about the sort of shift rather than a paradigm shift. Mm-hmm. It was more of a gradual adoption of new technology rather than the replacement of the old. What can you tell me about that?
Well, I don't think we should just throw out the old because it's old. It has a good solid base, and we're going to use it for a number of years. And we're always looking for new technologies that can make our lives better, help us solve more crimes, turn around faster, maybe do it cheaper and better than we are now. And those are all the things we want to look for. So yes, next generation sequencing is a bad term because there's always something next.
Yes.
That now generation or massively parallel sequencing or current generation may be a better way to think of it. And I don't think we're necessarily going to throw out everything we've done, and we should never do that. In fact, the good majority or portion of the technology is going to use the same technology we're using today. And I don't think this is a paradigm shift, this leap into something so dramatically different. It's just a new way of detecting the DNA, and maybe more like sequencing on steroids for our purposes, doing a lot more markers, a lot more information at one time, but not such a great leap in change, and more of a gradual change to another detector of DNA typing. We're going to try to use everything we can. And as I said, the first portions of the technology will not change. You still have to collect samples. You have to collect them in an efficient and effective manner. You still have to get the DNA off of whatever you collected it. And then you've got to do this thing called polymerase chain reaction, where we make copies of the DNA, millions and millions of copies in a test tube. That's all the same. None of that's going to change. So all that experience and knowledge and tools will be the foundation for the now generation sequencing, which will then allow us to look at more genetic markers at one time.
And there's current work and research on the improvement of the collection technologies, right?
Absolutely.
Improvement of purification technologies, improvement in the polymerase chain reaction technologies. And yet there's SNP information, there's mitochondrial information, there's STR information. What can you tell me about sort of where you see things moving in terms of what people are going to look for?
Yeah, well, you mentioned 3 different categories of genetic markers, and each one in themselves may have subcategories. For instance, SNPs. We just don't think of a SNP, which is just a change in the side of the nucleic acid sequence that we're interested in, and there's millions of those in the humans that can be exploited. However, there's SNPs for identity purposes, there's SNPs for ancestry, determine where your ancestors came from. Your ancestors came from a different place than my ancestors. We can tap into the genetic information. So if I found some bones at the crime scene, and unfortunately if they were your bones, which we hope not, and they were unknown, we could say this person comes from an Asian ancestry, or were my bones, they say from a European ancestry. We could also determine phenotype, hair color, eye color may occur. As I said, also SNPs for pharmacogenomics, that people who take drugs sometimes have adverse reactions based on their genetic constitution, and so this might help investigations.
On the pharmacogenomics piece, in terms of how you'd use that for forensics.
So you think of it as a molecular autopsy. About anywhere from half to about 1.5% of all autopsies, they can't determine the cause of death. About 2 to 6% they can't tell the manner of death. And there's actually a classic example of this, of a case where a 13-day-old newborn basically died of morphine poisoning. And as you imagine, 13-day-old was not out going collecting the drugs because couldn't get it on his own. So immediately there's an investigation looking at the parents, maybe a boyfriend, a family member, maybe some negligence in the hospital, and maybe some drugs were mislabeled. It turned out that the mother was given codeine for an episiotomy at birth, an analgesic just for pain. And if you know, codeine is metabolized in us to morphine. But she was an ultra-metabolizer. She had a double dose of the gene that encodes the enzyme for the metabolizing of codeine. So she was breastfeeding her child morphine at lethal doses. So again, by learning that she had this genetic predisposition to being an ultra-metabolizer, helps the investigation and turns it away from an infanticide to an unfortunate accident. So you knew the cause of death was morphine poisoning, but you didn't know the manner of death. So these kind of bits of information can help us in our investigations to go down. So SNPs, wide variety of those. The STRs are often used for identity testing. They are a foundation and they're very good for that. They're very good for interpreting mixed samples. And a lot of violent crimes or touch samples have multiple people contributing to them. And the mitochondria is a special, unique genome we have in our cells that without it we wouldn't be where we are today, as people call it, the genome for the powerhouse of the cell. But in it carries a lot of information that's useful for looking at maternal relationships, for missing persons or disaster identifications, to long distance kind of relatives that may be used as reference samples to help solve crimes. We've looked at it recently for human remains out of South Dakota, for instance. So we've done an interesting study with the Historical Society of Deadwood, South Dakota. And most of the people don't know anything about Deadwood, South Dakota, but it's a famed historical town from the Old West. Wild Bill Hickok, Annie Oakley, and all those people there, and established after the gold rush when Custer found gold in the area. And they had a cemetery there when they first formed. 2 years after that, they moved it. So about 140 years ago, this all occurred. And then just recently, they were building a retaining wall where the former cemetery was, and they found some remains. So now they have these 140-year remains, no information about them. And they asked us if there's anything we can do. So now we use the current generation sequencing and using all these new tools where we used STRs, mitochondria, and SNPs to help characterize this person. And we now know, consistent with anthropology, he's of European descent. He's very attractive because he has red hair and brown eyes. And I used to have red hair, and I still have brown eyes. And this is information you couldn't get beforehand. So you start to put at least some personality to the individuals of old times because we can tell more about them. And the future is going to be even greater and better abilities.
You're talking about hair, talking about eye. Do you think that facial reconstruction is potential in the future? Or do you think that it is a really tough task?
Well, it is a tough task. But then again, it's only trying to figure out information and getting there. We know that there's a high heritability on the way you look, because you, similar to your family members, identical twins are identical as far as we can see. And there are some features, but it's going to be a very challenging thing, because particularly for the development of the head, there's multigenic, as you can imagine, and that. But there's a lot of promise there, and there are some good work being done by a number of groups where they're using the ancestry, the gender of individuals to help reduce or constrain the morphology of the face and the prediction. And then if we get a few more markers, we might do better. But for our purposes, if we can get hair color, eye color, hair texture, soft tissues, the shape of the ears, the nose, the lips, the ones that you can't get off the skull, that can help the forensic artist better drive that. So if you found a skull in the desert and you want to figure out who this person is, that's difficult. Even with DNA, the people who may be the relatives of this individual may not be in your database. If they're not in a database, you can't solve it. But sometimes reconstructing the face and then putting it out to the media might get someone to say, oh, I know this person, and that. But if you reconstruct the face and it doesn't look anything like the person because you thought they had straight hair, because Europeans have straight hair instead of curly hair and the person had curly hair, could be just the thing that tips it in the balance by making an identification. And then you can do DNA directly on the person who comes in and the remains to get a very strong association, or refute it if it's not real.
Well, thank you very much for sharing today. Really enjoyed your talk.
That's my pleasure.